Hey everybody, welcome back to BIPN 140. So this next set of learning objectives will be all about channels. So these are some channels and how to investigate them using electrophysiology. We'll be using the voltage clamp technique that we just learned about and using it in different ways, like applying the voltage, different patterns of voltage and measuring slightly different things in order to investigate different aspects of how channels function. So electrophysiology, it's actually a very powerful technique, and there's a lot of things that you can learn with just, some wires and some circuits. And so we're going to get into that now. I do want to say that in these examples in these videos, we are going to focus on sodium channels. Sodium channels are really important for the action potential, and they're really important for epilepsy and for the case study that we're doing on GIFT Plus. But these techniques that I'm talking about today can be used to investigate any channel. They're very, very powerful, and people use them over and over again to characterize channels, to characterize mutations of channels, and to characterize different kinds of toxins that affect people and animals through interacting, poisoning these channels. So let's go ahead and dive in. So first up, we're going to talk about using electrophysiology to determine how the channel opens or what opens the channel. So this is called gating. So we have talked about leak channels. Leak channels open randomly. So we've talked about the potassium leak channels in the membrane. They open randomly. They're also known as rest channels. You can't do anything to make them more or less likely to open. They just open. On the other hand, I would say most channels are not like that. They are gated channels. That means they open under certain conditions. It's like a gate in a fence, right? It opens sometimes, when you open it, and it's closed most of the time. And so we have voltage gated channels, which we're going to be talking a lot about. We have ligand gated channels that can be opened by either external or internal stimuli, chemicals, and we have sensory channels, so like light gated channels, mechanical channels, thermal channels, all sorts of things. And so how do you know? How do you measure whether a channel will open to a particular stimulus? In other words, how do you measure whether a channel is gated by a particular stimulus? So the basic strategy is you use voltage clamping, and you voltage clamp the neuron and try the stimulus out, and you see if current passes through. And so, and on top of that, you usually kind of try different strengths of the stimulus, or you often try different strengths of the stimulus to just kind of confirm, oh, like if you have more of the stimulus or a stronger stimulus, there's more current passing through, which kind of makes sense, right? And so here's an example, the channel TRP A1. This channel, like many other channels, actually responds to two different kinds of stimuli, at least actually. So there are some chemicals that will open this channel, and it's also voltage gated. And so in this paper, they showed both of that. They applied this chemical AITC, and they applied it during this time when the bar is shown, and they did voltage clamping, and they saw that the current went up when the chemical was applied. So that implies that the chemical is causing the channel to open. That's why there is current coming through. The current is not zero, the current is coming through. This happens to be a positive current because of the nature of the ion that's going through. You'd have to do more work to figure out what ion is coming through. Okay, then they also looked at voltage over here. And so this is what they did. Kind of, if you see lines like this, that shows the voltage that was being applied in order to get the current. So these are the currents, these are the voltages. So in this case, they started at -80 and they tried different voltages. They tried, or actually they started at -70, that's the baseline, and then they tried different voltages. They tried going down to -80, holding it for a while, and then bringing it back up to -70. They tried holding it at -80 the whole time. They tried raising it to -60, -50, -40, all the way up to +200. And so there they held it to +200, and then they dropped it back down to -70. So they actually did many, many experiments here, right? So they tried each voltage probably multiple times, and that's how they got the current shown here. So this is actually summarizing like many different experiments and the voltages they applied during those many different experiments. Okay, and these are the currents they got here, and you see here that you see many lines here. So again, this is the results of actually many experiments. Each of these lines, including like down here, it looks thick because like many of the lines overlap. So each of these lines represents the current from one of these experiments. So probably the ones at the bottom here, those are the results of raising the voltage just a little bit, like maybe to -60 or to -50, whereas this one up here, that's the result of raising the voltage all the way to +100, 200. So you see here, this top here, you get a lot of current if you hold it at +200, and then this all drops off because this is where it drops off. This is where it went back to -70, so the current goes away when you drop the voltage away. So overall, this is showing that this channel is also voltage gated, and that the more voltage, the more you raise the voltage, the more current you get. So that is characterizing the gating of this channel. So this is from using that, you can make what's called an IV curve. And this is particularly important for voltage gated channels where how much voltage you apply matters. So you do an experiment like this, right? You'd try voltage clamping at many different voltages, and you would record the currents you get at each voltage. So this happens to be a voltage gated sodium channel. And one way you know that is because when you open the channel, the currents you get are negative. Remember that a negative current, remember this is voltage clamp, so a negative current represents an ion entering the cell, and sodium enters the cell most of the time when you open the channel. So that's what's going on here. And right, so each of these blue lines is the voltage that produces one of these black lines, which is the current. And you can kind of summarize that by making what's called an IV curve. So on this is down here, that is the voltage axis. This is the current axis. So it's saying for every voltage, what is the current? And I want to point out there's no time dimension. Okay. You see, like for this curve here, which is probably the biggest voltage, or the biggest current, like for this current down here, current changes over time. It kind of takes a bit to get to its peak, and then it falls rapidly. We'll talk about why it falls rapidly, but you just see now that it falls rapidly. So in order to make this IV curve, you just pick the peak current. So like this here, that would represent that dot there because that's the peak current for this particular voltage. Okay, so this is kind of showing you the peak, the IV curve shows you the peak current for every voltage. And yeah, basically it is summarizing right here the dependence of the current on the voltage. So you see if you try very low voltages, there's no current. If you try voltages around 0, you get a very strong negative current. And then if you go up to like +90, you start getting a positive current. Okay, so that is what this is showing. So this also, right, tells you about the gating. It tells you during what voltages you don't get current and what voltages you do get current. So this is again showing that this channel is voltage gated. It opens at high voltages. Okay. And just to reiterate that, right, like why is the current 0 kind of over in here? It's because the channel is not open at those low voltages. This is a voltage gated channel. So here is an example of how you can use this technique to understand how different kinds of mutations affect a channel. So in this case, there was a mutation, and this mutation is associated with some disease. I don't remember. Okay, and so this is the wild type. This is the mutant, and this is the both. And you can see with the mutant is the open circles. This mutant basically doesn't open. The current is flat the whole time. Actually, the authors said that perhaps the mutant channel doesn't even reach the membrane. That the mutation prevents it from going to the membrane. Whereas you can see before, the wild type has that strong voltage dependence, where it opens very strongly with voltage, and then it the current goes back up. We'll talk about why the current goes back up in kind of one of the future videos. Okay. Very interestingly, they also tried kind of the heterozygous situation, where both the wild type and the mutant are present. And then if you look at the IV curve, this heterozygous has a lower peak current than the wild type, and it sort of opens at a higher voltage than the wild type. So you can see the wild type starts opening at like -50 or so. It starts opening a little bit, whereas this heterozygote only really starts opening at like -40. So you can see here that there is a difference both in kind of overall how much current there is, and in the voltage gating. It is less sensitive to voltage. It needs more voltage to open than just if you had wild. One thing I'm often asked is if you look at a curve like this, or scientific data like this, like how do you know that these curves are significantly different from each other? Because there'll be situations, I mean, these curves are pretty far apart, but there will be situations where like the mutant and the wild type may be closer, a lot closer than this. And so one clue is that, well, first of all, in many papers, not always, but in many papers, the researchers will put stars in at particular time points to indicate significant differences. So there might be like stars here, here, and here to show that these are different from the wild types. These researchers chose not to do that for this particular figure. That is an okay choice. But if you see them, that's a step up, that it is significant. Another way to tell if you don't see those stars is that overlapping error bars mean non-significance. So in this case, the error bars are very far apart from each other. They don't overlap except around here. And so that is a clue that this is a significant difference. In other situations where the error bars overlap a lot, that would mean it's not significant. It might be a little bit different, but it's not a significant difference. Okay, well, that is all for this. I hope you kind of understand better how to use electrophysiology and voltage clamping to discover what gates the channel, so what basically makes it open, and how to use an IV curve to look at gating, voltage gating, and changes in voltage gating. All right, thanks so much.